What's up science killings and welcome back. In this video lecture we are going to discuss about ABO blood grouping, that is also called as ABO blood typing or blood matching. Determination of blood types A, B, AB and O is totally based on the expression of certain antigens on the surface of red blood cells or RBCs. So here we have the red blood cell or RBC. So if the gene on DNA codes for antigen A, so antigen A over here will be expressed on the surface of red blood cell and we'll say that the blood type is A. And if the gene on DNA codes for another antigen B, so antigen B will be expressed on the surface of red blood cell and we'll say that the blood type is B. In case if the gene on DNA codes for both antigens A and B, so both antigens A and B will be expressed on the surface of red blood cell and in that case we'll say that the blood type is AB. In fourth case, if the gene on DNA codes for no antigens, so no antigens will be expressed on the surface of RBC and in that case we'll say that the blood type is O. Okay, you might have heard that the blood type is positive, negative, like A positive, A negative. That is due to one factor and that factor is called RH factor. That was actually firstly discovered in rhesus monkey. That's why it is named as RH. RH factor has many antigens, but the most antigenic antigen in human being is antigen D. So in case of RH factor, the interpretation is totally different. That is, if RH factor is present on the surface of RBC, so we'll simply say that RBC is positive for RH factor. And if RH factor is absent on the surface of RBC, so we'll say that RBC is negative for RH factor. So let me draw over here the antigen. So this is the antigen and this is its Y-shaped corresponding antibody. So this is Y-shaped antibody. So if the antigen and its corresponding antibody, they are mixed together, so they will cause agglutination. That is also called as clumping. That is actually collection. That is actually the collection of separate particles like red blood cells into clumps or masses. That is called as agglutination. And that's why we call the antigen as agglutinogen, because this antigen has the capability to cause agglutination and we name the antibody as agglutinin. So if the agglutinogen and its iso-agglutinin are mixed together, so they will cause agglutination. Okay, in laboratory, to group the blood or to type the blood or to match the blood, a laboratory test is done. That is called as blood grouping test. In that test, we require two things. Number one, test sera, and number two, recipient's blood. So let's suppose we take over here one slide, correct? So we take one slide and on this slide we take three anti-sera, anti-A, anti-B and anti-D. And these anti-sera contain antibodies, the corresponding antibodies. So anti-antibodies and anti-B antibodies and anti-D or RH factor antibodies. So number one, we take these anti-sera and number two, we take the recipient's blood and we collect that blood at three different points. So once you mix them together, they will cause agglutination. And according to that agglutination, we will type the blood then. Let's suppose if agglutination takes place on A, so we'll say that the blood type is A because the RBC must have antigen A. That's why it has agglutinated with anti-A antibodies. Okay, before really starting the overall topic, I have written here at the top one very important rule to keep in mind, and that is that these blood type patients can donate their blood to these blood type patients. So blood type A can donate to A and B, and blood type B can donate to B and AB, and blood type AB can only donate to AB, and blood type O can donate to A, B, AB and O. And those blood types that are coming in negative can donate to both negative as well as to the positive, while those blood types that are coming in positive can only and only donate to the positive, not negative. So this is very, very important to keep in mind before starting the overall topic. So let's start from the very first example over here. And we imagine that, let's suppose there is agglutination on A. So it means antigen A is present on the surface of RBC. So we'll type the blood as, so if antigen is present, so we'll say that blood type is A. And you can see there is no agglutination or no RH factor. So it means RH factor is absent. So we'll say that RBC is negative for RH factor. So the blood type will be A negative. Okay, donate. What's the rule for donate for A? So A can donate to A and AB. So it means that blood type A can donate to A and AB. And what's the rule for negative? Negative can donate to both to the negative as well as to the positive. So A negative can donate to A negative and AB negative, and A negative can also donate to the A positive and AB positive. Okay, receive. This ABO blood grouping system and RH system both were discovered by Karl Landsteiner in 1901. Later on in 1930 he was awarded Nobel Prize as well. So this, according to ABO blood grouping system, according to Karl Landsteiner's law, there are 2 points. Number one, if antigen is present on the surface of RBC, so its corresponding antibody must be absent in the serum. Number two, if the antigen is absent on the surface of RBC, so its corresponding antibody must be present in the serum. So here, there is antigen A on the surface, so it will not have antibodies in the serum, anti-antibodies, while this person will develop antibodies against, so Y-shaped antibodies against B and antibodies against RH. So it cannot receive the blood from blood type B and patients having their blood in positive. So it can receive the blood from A negative and O negative. O negative will come in all cases because O negative blood type patient is universal donor. So we'll check that in the last case then. So here, yes, it's very simple. And let's proceed to the next example. And this time, let's suppose there is agglutination on A and RH. So it means there is antigen on the surface. There is A antigen on the surface. So we'll type the blood as A. And RH factor is also present. If present, it means positive. So we'll type the blood as A positive. Donate. Yes, the rule for A can donate to A and AB. So A can donate to A and AB. And positive, the rule for the positive is that positive can only donate to the positive. So A positive can donate to A positive and AB positive. Okay, receive. According to Landsteiner's law, so this person will develop antibodies against B. So it cannot receive blood from blood type having B, correct? So it can receive the blood from A negative and A positive, O negative and O positive. Why O positive? Because here in this case RH factor is present. It is the self antigen, so it will not make antibodies against it in the serum. So there will be no antibodies against RH factor. So that's why it can receive the blood from O positive as well. Okay, now let's come to the third example. In this case, let's suppose there is agglutination on B. So it means agglutination has taken place, anti-B antibodies and antigen B. So it means antigen B is present, so we'll type the blood as B. So it will be the blood type B. And there is no RH factor absent, so it will be B negative. So donate. What's the rule for B? B can donate to B and AB. And what's the rule for negative? Negative can donate to both negative as well as to the positive. So negative as well as to the positive. So B negative can donate to the B negative and B positive, and it also can donate to AB negative and AB positive. Okay, receive. So this person will develop antibodies against A. So Y-shaped antibodies against A, and also antibodies against RH, because these are the foreign antigens. So it cannot receive the blood from blood type A and also from the patients that are having positive blood type. So it can receive from B negative and as well as O negative, because it is universal donor. Okay, let's proceed to the next example. In this case, let's imagine that there is agglutination on B and there is also agglutination on RH factor. So it means if it has antigen B on the surface, so the blood type will be B, and RH factor is also present, so it means B positive. Donate. So what's the rule for B? B can donate to B as well as to AB. So yes, blood type B can donate to B and AB. And what's the rule for positive? Positive can only donate to the positive. So blood type B positive can donate to B positive as well as to AB positive. Receive. So this person will have developed antibodies against A. So it cannot receive the blood from blood type A. So it can receive the blood from B negative, and it can receive the blood from B positive. It can receive the blood from O negative, and it can receive the blood from O positive. Okay, now let's proceed to the next, and that is, let's suppose this time there is agglutination on A and there is agglutination on B. So it means both antigens are present on the surface of RBC, so we'll type the blood as AB. So what will be the blood type? AB. And RH factor is absent, so it means AB negative. Donate. What is the rule for donate? Donation of AB, blood type AB. So blood type AB can only and only donate to AB. And the rule for negative is this, that negative can donate to both, negative as well as to the positive. So it can donate to AB negative and it also can donate to the AB positive. Receive. This person will develop antibodies against RH. So it means it cannot receive the blood from those blood type patients that are coming in positive. So it can receive the blood from A negative, B negative, AB negative, and O negative. Okay, now let's proceed to the next. In this case, let's suppose there is agglutination on A and there is agglutination on B and there is agglutination on RH. So it means all three antigens are present on the surface of red blood cell. So if AB, both antigens are present, so we'll type the blood as AB. So what will be the blood type? Blood type is AB, and RH factor is also present, so it means AB positive. So donate. AB can donate to only AB, and the rule for positive is that it can only donate to the positive. So blood type AB positive can only donate to the AB positive. Receive. As all these three antigens are present on the surface and these are the self antigens, so the body will not make antibodies against them. So there will be no antibodies against these three antigens in the serum. So it can receive the blood from all blood types. So it can receive the blood from A negative, B negative, AB negative, and O negative. And it also can receive the blood from A positive and B positive, AB positive, and O positive. That's why we call this blood type, AB positive blood type, as universal recipient. We call this blood as universal recipient. Okay, let's proceed to the last two examples now. Okay, in this case, let's suppose there is no agglutination on A, B, but there is agglutination on RH. So it means there is no agglutination on A and B, so it means there are no antigens on the surface of RBC, and we'll type the blood as O. And you can see over here that the blood type is O, and RH factor is present, so it means it is O positive. Okay, donate. What's the rule for O? O can donate to A, B, AB, and O. And what's the rule for positive? That positive can only donate to the positive, not negative. So O blood type O positive can donate to A positive, B positive, AB positive, and O positive. Okay, receive. As this person has no self antigens A and B, so this person will develop antibodies against A, so Y-shaped antibodies against A, and anti-B antibodies in the serum. So it cannot receive the blood from A, B, and NB. So it can receive the blood from O negative as well as O positive. Okay, now let's we are having actually the last example over here, and that is O negative, the universal donor, and there's also my blood type, there's also my blood group. So in case of this, let's imagine that there is no agglutination on A, no agglutination on B, and no clumping on RH. So it means no antigens on the surface of RBC. So we'll say that the blood type is O, and there is no RH factor, so it means O negative. So what's the rule for donation of O? So O can donate to A, B, AB, and O. And negative blood type patients can donate to negative as well as positive. So it means O negative can donate to all blood type patients. That's why we call O negative as universal donor. Okay, receive. As this person has no antigens on the RBC, that's why it will have all antibodies against these antigens in the serum. So anti-A antibodies, anti-B antibodies, and anti-RH antibodies in the serum. So it cannot receive the blood from blood type A, B, AB, and it can only and only receive the blood from O negative. So it is a very rare case. So it is a very special case that O negative can only receive the blood from O negative. So this was the overall overview of blood matching or blood typing. So let me write it here in a simplified way in a formula, that blood matching is actually a laboratory technique or laboratory test in which you take the recipient RBC and test sera. So you rank these two. Okay, once you have typed the blood, and while you are just, before you're going to transfuse the blood from one patient to another, there is another method that is very, very important before you transfuse the blood, that is called as cross-matching. So what is the difference between cross-matching and blood matching? In cross-matching, actually you check the compatibility between donor's blood and recipient's blood. So you take the donor's RBC. So you take donor's RBC and you react that with recipient's serum. So these are the two very, very important formulae to differentiate between blood matching and cross matching. So these are the two very, very important formulae to differentiate between blood matching and cross matching. Why this cross matching is very, very important? To understand this, let me introduce another antigen, and that antigen is called H antigen. H antigen is actually the precursor of both antigens A and B. So let's suppose here we have the RBC and let's imagine this is the DNA, and if there is gene for antigen A, so antigen A will be formed by H antigen, and this A antigen will be expressed on the surface of RBC. So H antigen is actually the precursor, and if there is gene for antigen B, so antigen B will be formed by H antigen, and antigen B will be expressed on the surface of RBC. Remember, if H antigen is present but there is no gene for antigen A and B, so still there will be no antigen formation on the surface of RBC in spite of presence of H antigen. So the blood type will be O. In rare cases, this H antigen is present in all individuals, but in rare cases, H, A, B, all antigens are absent, and we see that the blood type is Bombay. So this is called as Bombay blood type because this was firstly discovered in KEM hospital in Bombay. That's why this blood type patient is named as Bombay blood type. And this is very, very much different from all blood types. So while you are typing the blood, Bombay blood type will seem like O blood type, while cross matching, this Bombay blood type will agglutinate with all other blood types. So I hope it made sense, and I hope it helped you a lot, and I hope you did enjoy. So if you did, please subscribe to my YouTube channel, like the video and share the video, and I thank you all. Thank you very much.